Bismillahirrahmanirrahim. Coming on to the cellular components biochemistry chapter, page 46, we'll cover most important MCQs from this page. I have left this cell cycle on page 438 to be explained in on that page because more MCQs are coming from that page and the same cycle is written there. We'll briefly cover the important MCQs. First is cell types. You will always get one MCQ, or most of the time you'll get one MCQ from it. There are three cell types based upon cellular division. One is the permanent cell that regenerate from the stem cells, that they do not regenerate from the previous cells present in the pool. They have to be regenerated from their stem cells, and the permanent cells remain in G0 phase. Examples are neurons. They don't divide, they stay permanent. Skeletal and cardiac muscles and RBCs do not have nucleus, so they cannot divide, and they are permanent ones. You can pick any of them, but if two are given, prefer cardiac over skeletal. It's up to you, but for all four, neuron, skeletal muscle, cardiac muscles, RBCs are permanent cell. Skeletal muscle do grow in size by hypertrophy, but not by hyperplasia, and we see cardiac hypertrophy as well. So these cells, if maximum can do is either hypertrophy but not show hyperplasia, and neurons do not hypertrophy and RBCs do not divide. So all types of examples in permanent cells. And then stable or quiescent cells, that they remain quite stable, and when stimulated they divide. And example is hepatocyte. We know that liver regenerate, so these cells are present where regenerative capacity is present, otherwise they are stable. And if stimulated by growth factor, for example hepatocyte growth factor 1 for hepatocyte is asked, it is not written here, so you can add on for convenience. An MCQ asked about stable cells is mostly hepatocytes. And then labile cells means they divide rapidly, and they are present in the bone marrow. For example, bone marrow or cells for blood, they keep generating blood cells. Here we have to see that bone marrow is labile, while RBCs, the terminal differentiated cells, are permanent cells. And gut epithelium is also labile, that it keeps shedding and keep growing. And skin is the same, so epithelia from skin inside the gut, all are labile cells. They regenerate after a few days if damaged. And then one thing confusing here is that we see that germ cells, they are labile, that they keep regenerating from the other germ cells. And stem cells are the cells which are pluripotent stem cells. For example, in bone marrow, it is bone marrow stem cells. And it is written here that in permanent cell that regenerate from stem cells, so we shouldn't confuse that stem cells are permanent cells. Stem cells are labile cells, and the cells regenerating from stem cells are a permanent cell, that they only regenerate from stem cells and then they are terminally differentiated. So sometimes it makes some confusion that stem cell is written above and bone marrow is written below. Bone marrow stem cells and stem cells are almost the same thing. It is written above for another meaning, that permanent cell divide from them, and below it is the labile cell or bone marrow. Then coming on to the two important organelles, we'll cover just two because they are overlapping: the rough endoplasmic reticulum RER and smooth endoplasmic reticulum. We know the difference between both. Rough endoplasmic is rough because of presence of ribosomes, rough and ribosomes. And ribosomes synthesize protein. This is often asked. And another MCQ asked is clearing of defective or misfolded protein. It is by proteasome. So either ribosome or rough endoplasmic reticulum for synthesis, and prefer ribosome over rough endoplasmic reticulum. And for clearing, that is the opposite function, it is proteasome of protein. And we know that mucin, it has protein because it ends with in for most of the protein. Mucus secreting goblet cells of small intestine will have abundant RER, that is an MCQ asked. So you have to keep in mind that this is important, otherwise we may skip this, that why it is more in mucus secreting goblet cells. Then a basophilia due to ribosomes attached. Rough endoplasmic reticulum will have ribosomes attached on them, and ribosomes are formed and they have ribosomal RNA. RNA is acidic because ribonucleic acid, and it will bind to basic die just like the nucleus. So nucleus and rough endoplasmic reticulum are basophilic if you look into the cell. And the question asked is basophilia in cytoplasm is due to, you can say it is due to ribosomes, and if not given, then it is due to rough endoplasmic reticulum. And Nissl bodies is rough endoplasmic reticulum in the neuron, and that synthesize peptide neurotransmitter. Peptide mean protein neurotransmitter, because rough endoplasmic reticulum synthesize protein, and specialized rough endoplasmic reticulum in neuron is called Nissl bodies. That is asked in MCQ. And the MCQ asked otherwise is that the Nissl bodies are lacking in axonal hillock region, because here the impulse starts and neurotransmitters are synthesized in the body, not the axonic hillock or the terminal portion of the cell body. Nissl bodies are RER of the neurons, and they are lacking in the axonal hillock. So both are MCQs. Then smooth endoplasmic reticulum, you have to remember, as for steroid and as for smooth, they synthesize steroids. So for synthesis of protein it was rough endoplasmic reticulum, for steroid it is smooth endoplasmic reticulum. And just following the green sequence, coming on the other side, steroid hormone synthesis is taking place in adrenal cortex, because we know the all the adrenal cortical hormones are steroids, and gonads are rich in SCR. Why? Because leading cells that produce testosterone, that is a steroid hormone, is synthesized in the gonads, so they are rich in SCR. So MCQ asked is leading cells synthesize testosterone, and they are rich in SCR. So you have to jot down this from page 6 to 8 to here. You can go to that page to understand it better. And the other color coding I have done is that smooth endoplasmic is the organelle which causes detoxification of drugs and poisons. And this specific detoxification is mostly taking place in the liver hepatocytes. Just steroid synthesis mostly in the steroids hormone sensing area like adrenal cortex and gonads. Detoxification is mostly liver function. And MCQ asked is it is in the zone 3 pericentral zone, having highest P450 enzyme involved. And you can refer to page 367 for this to understand it better. Another confusing MCQ asked is alcohol detoxification or ethanol. It is given on the next page. Alcohol detoxification is peroxisomal task, and peroxisome shouldn't be confused with proteasomes that are used for defective protein or misfolded protein. So organelles overlap with each other, but you have to keep in mind the differences between them. RER and SER, two important organelles with very important functions for body, and the location of the cells involved having highest RER in GIT and SER in gonads is asked. Thank you.